Hi guys, it's Rob from Royal Balls and it's time for my October roundup. We have a lot to get through in this edition and I'm actually going to call in Mr. DNA to talk about some cellular level genetic stuff that I've been seeing on social media. But first of all, what's going on in the snake room? Well, I have stopped pairing. I'm not breeding anymore and I will not be breeding again until the market picks up. I am full and have no more room. But don't worry, we still have snakes that are due to lay, we still have clutches in the incubator, and we will still have a lot of stuff to talk about. Alright guys, I just wanted to show off my Enchi Super Scream Clown. So it's Enchi, Super Pastel, Fire Vanilla, and it's a visual clown. It has taken its first meal. And it is super bright. This is effectively a triple homozygous morph. It is a visual clown, so homozygous for clown. It's super pastel, so homozygous for pastel, and it is also fire and vanilla, which is the allelic combination of those two genes, so effectively again and acts like a super or homozygous. Now I am going to do a video for you guys on distinguishing fire from vanilla, because this came from a clutch where obviously both fire and vanilla are present, but the whole clutch is not eating yet, so I did want to wait until the colours come in a little better and they're eating and established before I do that video. So it is coming. Now ARP has just produced what we thought was a super pastel highway clown, so again a triple homozygous, super pastel, homozygous for pastel, clown, visual for clown, homozygous for clown, and highway, the ALS combination between gravel and yellow belly. But he made a second one, and the second one can't be a super pastel because that isn't possible in the pairing, and it is identical. So actually it turns out that it isn't a triple homozygous, but it is a phenomenally pretty snake, the best snake that I've seen this year. So just compare this guy with the pastel highway clown that ARP has produced. Just look at the colours on this guy. Wow. As far as the YouTube videos are going, I'm rapidly approaching 4K subscribers, which is absolutely awesome. I have noticed that YouTube is taking away from me control over the ads that show on my video. My videos are monetized and YouTube do put ads on them, So let me know in the comments below if the ads are getting excessive. I still have some control over what ads go in and where the ads actually go in the video. I need to have ads in the video in order to make money. Just to give you an idea of how much money I am making. You can see in my videos list that I am making videos very regularly. I have almost 4K subscribers and most of my videos get anywhere between 500 and 1,000 views. That translates to about $100 a quarter that YouTube pay me. I have a threshold payment of $100. Typically when it gets to about 120, they will send me a check, but it is taxed at source in the US at 25%. So $120 usually means I get about 85. Despite the fact that I'm retired, have no other income, and I am not a US citizen, I am paying US income tax even though I'm below any threshold income. They tax me at source, they send me an income tax form, and I am supposed to claim that back from the Malaysian authorities. Yeah, right. Do let me know, guys, if the adverts are becoming excessive. It's not really monetarily valuable to me, and if necessary I will delete adverts from the videos so that you guys get a better viewing experience. Alright, enough about what's going on in the snake room. Let's talk about what's happening in the social media world. And I've been watching with a great deal of interest the latest episodes from Rare Genetics Incorporated, RGI, who are doing all your shed tests for you, including the DG stuff, and there's a lot of new info coming out in the last few episodes. So I'm going to bring in Mr. DNA and we're going to talk about some of the things that RGI are talking about which are related to cellular level genetics. Don't worry, we're going to keep it simple. You know Mr. DNA is going to use his pipe cleaners and you'll all be able to follow this easily. So let's get after it. Come on in, Mr. DNA. Alright, what I've got here is a bunch of chromosomes and a bunch of genes, and this is pretty much what it would look like inside the nucleus of any cell within our ball pythons, a complete mishmash of different chromosomes and genes. So the first thing that I want to talk about, the first thing that I'm hearing a lot about that seems to be gaining traction, is temperature sex determination in ball pythons, and many people are now claiming that their incubator temperatures affects the sex ratio of the babies. Running warmer temperatures produces more females. Okay, let me start off my discussion by saying that Rare Genetics Incorporated are doing a lot of the genetic testing on morphs of ball pythons, and they're identifying various genes within your ball pythons, and most of you I think are comfortable with the science behind that. Most of you are comfortable with the concepts. So Rare Genetics Inc. are also offering sex determination from shed testing. Hang on a minute. If they can determine sex from shed testing in exactly the same way as they can determine what genes there are in your snake, doesn't that mean that the signature for sex is contained in the chromosomes within the nucleus of that cell? Well, yes it does. What they're actually looking for within the cells of our ball pythons is female XX chromosome, male XY chromosome. So if these chromosomes are present in the genetic code, does that not mean that as for all the genetic mutations, that code has been written into the cell at the time of fertilization, which means that temperature has nothing to do with it? Yes, that would be the implication of being able to determine sex from shed tests. Now, another line of evidence that we already have in ball pythons, all those of you sending in your shed tests to identify genes, I don't think anyone is claiming that they can switch on or switch off Enchi or leopard or fire or any of the other genetic mutations in their incubator. Once that genetic code is fixed at fertilization, the snake is going to be an Enchi, and there's nothing you can do to change that. So why would we believe that we can change a Y chromosome, which is determined at the time of fertilization, into an X chromosome in our incubators? The answer is we can't. The proof of that is actually in the banana sex link. The evidence is staring us right in the face. So here is our male ball python. It is an XY animal. Fortuitously, it's exactly the same as humans. Humans are also XY chromosome sex determination, with females being XX and male being XY. So because our ball pythons receive half of their genetic code from mum and half of their genetic code from dad, this means that they are diploid animals, and every single chromosome within the nucleus has an identical counterpart from the other parent. So chromosome 1 will have an equivalent chromosome 1 from the other parent. Chromosome 9 will have an equivalent chromosome 9 from the other parent. They are doubled up throughout, and ball pythons have 18 pairs of chromosomes. Humans have 23 pairs of chromosomes. So a ball python has 36 chromosomes in total, a human has 46. So we're separated by only 5 pairs of chromosomes. Isn't that an incredible thought? So here we go. Your female is XX, pink pink. Your male is X and Y, pink and black. And you will notice that in the female, the 2 chromosomes here, XX, are still identical. These are known as homologous chromosomes, just the same as every other chromosome has an exact counterpart from the other parent. The only difference is in the male. X and Y chromosomes are not the same. They are not homologous. They are not identical. So we've long known that the banana gene is sex-linked. Our male, our XY animal, is the animal that determines sex, and depending upon whether your male is a male maker or a female maker, all the bananas he produces will be either male or female, unless there is a rare crossover event. So here we go. Here is your male XY animal. He carries the banana gene on one of those chromosomes. The banana gene resides on either the X or the Y chromosome, just as any other genetic mutation will reside on any of the other chromosomes. It just so happens that banana resides on the sex chromosomes X and Y. It's the only gene that we know of that does this. So if your male is a male maker, the banana gene is carried on his Y chromosome and is not carried on his X chromosome. So I'm going to simplify gamete production here in terms of meiosis. In meiosis, any cell that divides is going to double up and then split into 4. But for the purposes of this demonstration, we're just going to use 2. So what happens here is that when your male produces his gametes, they are haploid. There is only 18 chromosomes, 1 full set, not 2 full sets. He's going to pass on 1 full set of his chromosomes in his sperm, and that will combine with 1 full set of chromosomes from the female. So here is your male. Every time he passes on a Y chromosome, he passes on the banana gene with it. Every time he passes on an X chromosome, he is not passing on the banana gene. This is a typical male maker. A female maker is the other way round. So a male banana who is a female maker, every time he passes on a Y chromosome, he will not pass on the banana gene. Every time he passes on an X chromosome, he will pass on the banana gene. So X combined with an X from the female is going to produce a female banana. Y combined with an X from the female in the case of a male maker is going to be a male banana. Now, normally in any other genetic mutation that resides on a different chromosome, when these chromosomes line up in preparation for division to make haploid cells, you can see that the locus for any particular genetic mutation lines up exactly with its counterpart from the other parent, and this means that it's quite easy for pieces of the genetic code to jump from one chromosome to the other. And this means that little pieces of dad mix with little pieces of mum to produce individual haploid cells which are unique. They are neither a direct copy of mum or a direct copy of dad, and this is what we want in sexual reproduction. We're not cloning, we want the offspring to actually contain the best of each of the qualities from mum and dad. Sometimes they don't, they contain the worst qualities from both, but generally speaking we get a variability that allows for evolution to occur, that allows for a species to adapt to new environments, because some of the offspring are going to be better suited to that environment than others. So normally for chromosomes 1 through 17 in our ball python, the genetic material is free to interchange between the two chromosomes and therefore could be present in any combination in any of the offspring. That is not true for the banana gene, which resides on the XY chromosome. When the cell divides to produce sperm and the X and the Y chromosome line up, because they are not homologous, they are not identical, the locus for any particular gene does not line up with the other side, and therefore it is almost impossible for the banana gene to jump ship. It cannot move from the Y chromosome to the X chromosome because they don't line up, and it's very difficult for that gene to make the jump. It does happen occasionally. About 5 to 10% of the time, male maker bananas will produce a female banana. So it can happen, I'm not ruling it out entirely, but the fact that in the majority of cases our male makers produce male bananas and our female makers produce female bananas, that is determined at the time of fertilization. That means that both the banana gene and the sex of the animal is determined at fertilization, not in the incubator. Okay, so when we look at chromosomes and genes, all chromosomes and genes do is control the production of hormones, which affects the production of certain proteins. So the chromosomes themselves are switching on and switching off hormones to produce specific types of protein, specialized cells, during the growth of the embryo. You can think of this genetic code, each chromosome, as a computer program and each gene as a subroutine. The computer program can call that subroutine each time it needs a specific type of cell generated in order to grow a baby snake from a bunch of cells. That's all chromosomes and genes do. They switch on and switch off hormones. Now, in temperature sex determination, the animal usually doesn't have a sex chromosome composition. Generally it is indeterminate, and that is because it's not the sex chromosome that is controlling the hormones switching on and off. It's temperature. Temperature switches on and off the hormones, and that produces males or females in temperature sex determination. But there is almost always a threshold temperature, below which you're going to get all one sex, above which you're going to get all of the other sex. There is very rarely any mixing in temperature sex determination because the temperature control is absolute. You're either above it or you're below it, which means that mixed clutches are rare in temperature sex determination. I think even people who are claiming that temperature of their incubator affects the sex ratios are still getting mixed clutches. They're not getting clutches of all one or all the other, which is how temperature sex determination works. Typically in temperature sex determination, the sex of the animal is determined in the middle third of incubation. So in the case of our ball python, if temperature is having an effect on the gender of the babies, then fertilization occurs 50 days before the eggs are laid. The eggs are in the incubator for about 55 days after that, so let's say 105 days, which means that sex is going to be determined, if it's the same as every other temperature sex determined animal, in the middle third of incubation, which for our ball pythons is between day 30 and day 60. Eggs are typically laid 50 days after fertilization, so even if temperature is having an effect, by the time the eggs are laid, the sex is almost certainly predetermined. We're already right on the cusp of the middle third of incubation, and by the time our eggs go in the incubator, it's too late to change it. So why do we feel, what could be fooling us into thinking that our incubator temperatures are affecting the gender of the offspring? Okay, here is our male sperm and here is our female sperm, and remember, it's the male that determines sex. He is the heterogametic sex. He is the animal that contains the XY chromosome. Females are XX, so all their eggs are all X chromosome, and they're waiting for the male sperm to join and either provide a Y chromosome or an X chromosome to determine the sex. If ball python sperm, like just about every other animal that produces sperm on the planet, is actually different, the male sperm swims faster but doesn't last as long as the female sperm. The male sperm is more sensitive to environmental temperatures. The female sperm is more robust and generally is less sensitive to environmental conditions. What typically can affect our sex ratios is, for instance, our hotspot temperatures. If our hotspot temperatures are too hot, the male sperm gets killed and the female sperm survives, and therefore when the male mates with the female, we're already skewing the likely sex ratio towards the female side because we've killed off the male sperms by hotter temperatures. If we use cooling, or if our environmental conditions have nighttime temperatures with quite a drop, it might be that the sex ratios are influenced by the temperature drop and we're favouring male sperm now over female sperm. This is even before your male has mated with a female, when he is generating his sperm. So conditions such as the type of heat mat that you use, the type of material that your tub is made of that has different conductivity, your environmental conditions, your nighttime temperature drops, all can affect the sex ratio of sperm. When a male produces sperm, they're always 50-50. That's the way the mechanism works. But that doesn't mean that before he meets up with a female, some of those sperm have succumbed to less than ideal environmental parameters, and therefore there are more female sperm or more male sperm. So unless you eliminate all of those variables, it's very difficult to say from your incubation temperature results alone that it's incubation temperature that is affecting the gender of the offspring. Likewise, When you pair your snakes, if you pair once a week, you're forever replenishing the sperm that the female is storing, which means that it's more than likely that the faster swimming sperm are going to be available in sufficient quantities and swim fast enough to meet the eggs that the female is generating at the time she ovulates. If you pair just once a month, many of the male sperm will have reached the top end of the oviduct, but there's no eggs there to fertilize, and it's the female sperm that lasts the month when all the male sperm are dying out. So if you pair more frequently, you're favoring males over females. If you pair less frequently, females over males. That's another variable that we need to account for before we can provide any data on incubation temperatures affecting the gender of the offspring. Now, if you pair the week before an ovulation, or you pair 2 days before an ovulation, again, because of the different swimming speeds, you're favouring males over females or females over males. The closer to ovulation you pair, the more you favour males. The further away from ovulation you pair, the more you favour female sperms fertilising those eggs. Again, another variable that is going to affect the gender ratio of individual clutches even before the eggs are laid. Never mind incubation temperatures affecting them. So there are a host of variables that are potentially affecting the gender ratios of our clutches, not incubation temperature. Sex is determined at the time of fertilization, and any skewing of sex ratios is much more likely to be down to our husbandry, our pairing frequencies, and other external factors. And I can hear guys saying now, yes, but 5 years ago I was running my incubator at cooler temperatures, and today I'm running them at warmer temperatures and I'm getting much more females. Yes, but 5 years ago you weren't using ultrasound to time your pairings. Using ultrasound reduces the frequency of pairings and favours females over males. It's another factor that we didn't consider in sex ratios that invalidates data on incubation temperatures. So if you are collecting data from your incubator temperatures and sex ratios of the offspring, do bear in mind the multiplicity of other factors, some of which are operating months in advance of eggs even being laid. It's so important to have proper control studies and eliminate the other variables that may be affecting the outcome. Otherwise, your conclusion is not going to be valid. Now, looking at my sheet of stuff that's been going on, I have been following Rare Genetics Inc. and their weekly updates, and there's some interesting stuff that's come out of some of their videos in the last few episodes. The first one is that they have tested a super leopard. They have tested and proved out a homozygous leopard which has been bred, and all the offspring were leopard. I do not know why this has come as such a surprise. Anyone that's followed any of my genetic series will know that I've long said that any gene can exist in heterozygous or homozygous form. Ball pythons are diploid animals. They have 2 sets of homologous chromosomes, which means there are 2 slots for any particular genetic mutation. Every single gene, whether it's dominant, incomplete dominant, or recessive, has 2 available slots. Look at it this way. Is there such a thing as a female leopard? A female leopard would be the heterozygous form, 1 copy of leopard, and here is her genetic composition. And we know that female leopards certainly exist, and female leopards can certainly pass on in their eggs a copy of the leopard gene. So yes, female leopards exist, and yes, female leopards can produce leopard offspring. Do male leopards exist? Yes, of course they do. And when you breed male leopards, can they pass leopard onto their offspring? Yes, of course they can. Half of the babies from a male leopard will be leopard. So why then, if we breed leopard to leopard would we think that this genetic relationship, the homozygous form, is not possible. Half the genetic material comes from mum, half the genetic material comes from dad. Both of them are leopards. This is homozygous leopard. Let me repeat that for everyone. Every single genetic mutation that we know about can exist in heterozygous or homozygous form. And yes, that includes spider and champagne. There are 2 slots on the genetic code for both spider and for champagne. And it is possible, if you breed spider to spider, that you will get a superform, with the caveat that spider to spider, champagne to champagne, if you create the superform, it is lethal. The eggs never make it and they die in the incubator, so we very rarely will see a super champagne or a super spider. I personally have seen a super spider, but it died after a couple of months out of the egg. So let me give you my definition of what dominant, incomplete dominant and recessive genes are. And this takes us into the next subject I want to talk about, which is phenotype versus genotype. They are not the same thing, but we have artificially superimposed them. We often refer to a phenotype as being a certain genotype, and this is not always the case. So a dominant gene is one in which the heterozygous form gives us an almost complete phenotype expression of that gene, and adding the second gene to it to make the homozygous form does not change the visual appearance of the snake. Now that is, strictly speaking, not true. It is possible to distinguish a leopard from a super leopard, but it can be very subtle and depends very much upon what other genes are in the mix. So a dominant gene, for instance pinstripe or leopard, would look pretty much the same and express pretty much the same in any combo as the heterozygous version. So 1 copy gives you the full visual expression, 2 copies does not change it a great deal. For an incomplete dominant gene, pastel would be the most familiar example to most people. 1 copy of pastel, the heterozygous form, has a visual expression. We can see it expressed in the phenotype. 2 copies of pastel, the super pastel, or the homozygous form of pastel, gives the full visual expression of pastel. It looks different to a single gene pastel. There is a visual difference and we can tell them apart visually, and that is the only reason that we make the distinction between dominant and incomplete dominant. Genetically, they can both exist in heterozygous form and in homozygous form. Contrast that with what we call a recessive gene, where 1 copy has no visual expression, and I say that reservedly. I'm going to come back to that in a moment. No visual expression in the heterozygous form. We need the homozygous form of a recessive gene in order to see the visual expression. So this female here is het for pied. It's actually pastel het for pied, but she is heterozygous for the pied gene. 1 copy of pied, which means that she is not expressing the pied phenotype. She does not look like a pied. Whereas this female here is a visual pied. She is homozygous for the pied gene, 2 copies. So this is what we call a pied, and this is what we call a het pied. Genetically, 1 is heterozygous for pied and 1 is homozygous for pied. This is pastel. It is heterozygous for pastel, only 1 copy of pastel. This is a super pastel, homozygous for pastel, 2 copies of the pastel gene. You can differentiate the 2 based on the head stamp, and the much busier pattern on this guy here. So, pastel, super pastel. So if this is het pied and this is visual pied, why is this not het pastel and this pastel? Because genetically the configuration is the same. Heterozygous, homozygous, 1 copy, 2 copies. This is an artificial nomenclature that we have produced because 1 copy of pastel has a visual expression. The full phenotype of pastel is this one, the homozygous one. If it's a recessive gene, we call this one het and we call this one visual. Pastel is nothing more than heterozygous for super pastel. Genetically, heterozygous, homozygous in a recessive or an incomplete dominant or even a dominant gene, it's the same genetic configuration. Now, I remember a long time ago when I first started making videos with Richard at Predator BP, I did a video on het markers, and I was told that unless we've been doing it wrong for the last 30 years, there is no such thing as a marker for hets. Well, I'm not going to tell you that we've been doing it wrong for 30 years. I'm simply going to tell you that our knowledge of genetics has improved tremendously in the last 30 years. And every single gene, whether it is heterozygous or homozygous, will have some sort of visual expression. So het markers are real. The heterozygous form of a certain gene may not express very strongly, but if we know what to look for, there will always be some expression. How could there not be? It's a genetic modification of the original DNA. How could there not be a change in what the animal looks like? Now, historically, we've been more than happy to use certain genes as visual enhancers to our combos. Fire, for instance, or yellow belly, particularly in clowns, we've used the heterozygous form of these genes, which are notionally described as incomplete dominant genes. We have used them as enhancers in our morphs. So, 1 copy of fire added to our genetic morphs makes the animal brighter. We do not want to make the super form of fire because that's a white snake and we wouldn't see anything else, but we're quite happy with incomplete dominant genes to use the heterozygous form of that gene as an enhancer. We're more reluctant to use the heterozygous form of a recessive gene as an enhancer. And why would that be? Well, historically, it's because we believe that the heterozygous form of a recessive gene has no visual expression, so how on earth could we use it as an enhancer for any particular morph? Well, I'm here to tell you that the heterozygous form of many recessive genes can also be used in exactly the same way. Lavender albino can be used as an enhancer for brightness in the same way that we would use fire, in the same way that we would use yellow belly, in the same way to some extent as we would use desert ghost. A single copy heterozygous form of lavender albino will enhance the look of any morph. You can use the heterozygous form of a recessive gene as a brightener or enhancer. This is a firefly het lavender. It is an adult female. I picked her out fairly easily from a clutch of Pos het lavenders because she was really bright, and she has retained that brightness right through to adulthood. Superfly het lavender. 2 doses of pastel and no sign of this guy browning out. Again, 3 years old, and it's the het lavender that is keeping this bright as a new pin. We are using het lavender as an enhancer. Yes, we do not want to make the homozygous form because that would be a lavender albino, and perhaps that's not our objective. But by the same token, when we use fire, we also don't want to make a super fire because that's a white snake and that would not be our objective. So using recessive genes versus incomplete dominant genes as enhancers is exactly the same thing. We do not want to make the super form, we want the heterozygous form for use as an enhancer. What else has happened at RGI? Well, they have announced that several genes that they are looking at have turned out to be exactly the same gene. We've known for years that there's been duplication in the industry. Coral Glow and Banana are the same gene. Mystic and Phantom has been announced by RGI as being the same gene, genetically no different. And interestingly, Butter and Lesser genetically are exactly the same. And I know that many of you are going to say, but Butter and Lesser are not the same. They look a little bit different. Yes, maybe they do. We should not confuse genotype with phenotype. We should not confuse the genetic signature, which is built into the DNA, with how an animal looks. And here I'm going to use Orange Dream as another example, and RGI again have hinted at this. We already know from Desert Ghost that Desert Ghost is polygenic. There is more than one gene involved in the phenotype for Desert Ghost. I have noticed over the years of working with Orange Dream that Orange Dream is highly reactive. It reacts differently with many different gene combinations. Some of them so strongly you would swear they're allelic when they're actually not. It is more than likely that Orange Dream is reacting in different ways depending upon what other genetic code is in the nucleus of the cell, and these could be totally unrelated genes. They may not have a name, they just accumulate in our collections to enhance the look of Orange Dream when you breed Orange Dream. So here's what I think is going to happen. RGI are on the brink of announcing that high intensity Orange Dream is nothing more than Orange Dream. It has a kicker with it, it has an enhancer, there is another gene associated with Orange Dream that allows it to express as high intensity Orange Dream. I think there has been a plethora of Orange Dream derivatives coming out onto the market, and I think you're going to find that they will be exactly the same. It is just Orange Dream, but the rest of the DNA contained in that nucleus gives it a slightly different look because it is so reactive. So it'll be Orange Dream with A is high intensity Orange Dream. Orange Dream with B or C or D will be one of the other derivatives of Orange Dream. This is where we could start to have a discussion on line breeding. Line breeding is breeding the best of the best back to the best of the best, and typically the snakes are related. In this particular case, because the gene pool could be in unrelated snakes and these other additional enhancing genes are floating around in the collection, it is not necessary to breed related snakes in order to improve the phenotype or the visual expression for that particular genetic morph. And this is where I think Butter and Lesser have gone their separate ways. Coral Glow and Banana have gone their separate ways. The base gene is still exactly the same. Same thing with Orange Dream. So what happens when a particular person works repeatedly with Orange Dream is that over the years his collection will accumulate all the other genes that make Orange Dream look fantastic. You buy one of those fantastic looking Orange Dream derivatives from a well-known breeder, you breed it to your own stock, and what happens? You get some pretty average looking Orange Dreams. Some of them a little bit better than others. Typically, if you breed high intensity Orange Dream, you are not going to get all high intensity orange dreams. You may get one or two that approach that, but you're never going to get back to the original unless you repeatedly breed back the best of the best and accumulate the other genes necessary in the genotype in order for that orange dream to express in the way that we hoped it would. ARP is a very good example of how this works because his collection does not have an infusion of new genes all the time. He has to make do with what he's got. So over the years, he breeds, and you would call it line breeding, and produces some animals with a phenotype that look very much better than the original phenotype from the original genetic infusion. And this is because he's concentrating all those additional genes in the collection as a whole. Breeding brother to sister and daughter to father does not necessarily give you the best looking animals, and in fact ARP's trademark bumblebee calicos, the best results come from breeding third, fourth or fifth generation animals back to the original great grandparents. They're the ones, those specific snakes are the ones that have the right genotype to allow bumblebee calico to express at its best. So line breeding does not necessarily involve breeding related snakes. Now I am waiting for RGI to confirm this story. I've been suspicious of Orange Dream for a long time, and I think that this genetic testing will certainly help to identify what we're actually dealing with and how best to deal with some of these genes. But we're finding now that with this polygenic condition allowing for different expressions of the base gene, that perhaps adding infusions of new genes is not the way to go with these genes. We need to be considering a different way to breed which tries to concentrate those additional kicker or enhancer genes that work so well with some of these genetic combinations. We're certainly going to have to do that with Desert Ghost. Now another announcement that was made by RGI is that DGC, the long-awaited DGC to complete the picture, is present in 84% of all visual DGs that were tested. The final results have yet to come, but it would not surprise me if DGC is actually on a separate chromosome, is a completely separate gene, and DGC can exist in both heterozygous and homozygous form, and it is just one of the components that go towards making a snake that looks like a visual DG. The phenotype and the genotype for DG are going to be different. When we talk about visual DGs, the phenotype, we know what we mean, but it does not imply a specific genotype. Now, I'm hopeful that as genetic testing progresses, that a lot of this duplication of names, Butter, Lesser, Coral Glow, Banana, Mystic, Phantom, here's a big one. This is going to be controversial for you. I am waiting for RGI to announce that Asphalt and Gravel are the same gene, genetically no different. Last year I produced some leopard freeways. This year ARP has produced some leopard highways. Guess what? You cannot tell them apart. So all these pavements and freeway, highway combinations are actually nothing more than the superform. It's the same gene. I do not know that for certain. I'm just being controversial. I'm waiting for RGI to announce that. Now obviously Asphalt and Gravel are different to Yellow Belly. Yellow Belly in its superform gives an ivory. Asphalt and Gravel in their superforms do actually give a visual superform, but it's the combination of Asphalt and Gravel. Can we actually say that they're any different to a super Asphalt or a super Gravel? So Asphalt and Gravel, watch this space. Are they the same gene or are they actually different? I know where my money sits. Okay, so we've covered that ground. A certain phenotype or visual expression does not necessarily imply a certain genotype. In the past, we have used them as synonymous, and we now know that they no longer are. And I think that Orange Dream, rather than Desert Ghost, is probably going to be the genetic mutation that illustrates this the best. Depending upon what other genes you put with Orange Dream, we are ending up with a plethora of what people are calling new genes. They're not. They're simply phenotypical expressions of a different genotype, of which Orange Dream is just one of the genes involved. Alright, I've had some very interesting conversations on other aspects of genetics in our ball pythons. For instance, eye colour. I had a very interesting conversation with Carl over at North Coast Constrictors, who has observed that eye colour seems to be related to certain genetic combinations, and this is indeed true. Eye colour is genetic. A specific eye colour is related to a specific genotype. However, when you start to mix genes into your snake that have axanthic, amelanistic, leucistic trends, you will also affect the colour palette of the eye, even though the genotype from the base genetic morphs is the same. So your dark morphs and your light morphs will express different colour palettes in their eyes based upon the amount of melanin that they have in their skin. This is exactly the same as humans. Different races with different skin colours may have the same genotype for a certain eye colour, but it expresses differently. So yes, eye colour is genetic. Yes, you can use eye colour under certain circumstances as a genetic marker. And we've long known, for instance, banana. When you shine a light into the eye of a banana, if you're not sure that you actually have a banana because you've covered it up with, say, a leucistic snake, you can still tell that it's banana because the eyes will shine back red. So genetically, yes. So here we have a leopard mystic. This is part of the leucistic complex. It is mystic and Mojave, and you will see that she has blue or bluish silver eyes. This is almost certainly a function of her genotype. This female spider has greenish eyes, but if we put an axanthic gene into the spider, the axanthic gene will remove the yellow colour from the green eyes, and that spider complex will have blue eyes. Other features that are possibly genetic: head shape in ball pythons. You've all seen ball pythons with blunt heads, with very long snouts. You've seen every variation of proportion in heads of a ball python, and yes, that's also genetic. It comes along with the genetic code when we breed our morphs, and we often don't pay attention to it, and it's not of any particular value. It's of casual interest only, but it is genetic. Okay, can you see how short and fat the head on this baby cypress is? This is yellow belly cypress, but this female has a particularly short, fat head versus this guy here, who has a much longer, more pointed head. Let me see if I can give you the comparison. Look at the difference in the head shapes. This one in my right hand here has a short, fat head. This one has a much more aesthetically pleasing pointed snout. So again, that is genetic variation. it can be passed on, and you can breed for a certain head shape in your ball pythons. No doubt that you can affect the head shape of your ball python depending upon what gene pool you are using when you breed your snakes. Here's one for Acacia, if she is watching this video. Temperament. Can we breed temperament into our ball pythons? Well, given that we've bred dogs from wolves, and wolves are typically fairly aggressive, it is most likely that to a certain extent, yes, you can breed for temperament. Now, what I would say with ball pythons is that your base morph, your starting canvas, is actually a fairly passive sort of snake. And there are actually other factors involved in the way your snakes behave, mostly down to the husbandry and the way that we treat them, that will allow them to develop adaptive behaviour. So for instance, people say that if you handle your snakes very often, that they become accustomed to handling. I have done videos on that, and yes, there is actually an effect. Not all of it is good, and I do not actually recommend handling your ball pythons frequently. Handling infrequently puts less stress on the animal, and overhandling can certainly change their demeanour, and they get very grumpy. They will often also go off food if we overdo it. Now, from a genetic standpoint, can we breed calm snakes? Well, given that a ball python is already quite calm, I have seen some examples in my collection where we've actually made it worse with a particular gene stack. I have a champagne which is overly defensive, overly stressed at all times. His father was the same, his grandfather was exactly the same. Here is my leopard champagne, and just look at this guy. He is absolutely wired. He has been like this all his life, extremely defensive. This guy does not like to come out and free roam. He does not relax. His father was the same, and his grandfather was the same. This behaviour is almost certainly related to his genotype, and in this particular case, it's the champagne gene. So genetically, yes, we can make it worse. I'm not sure that we can make it better. However, I'm open to your own observations on that, but certainly a lot of ball python behaviour is not genetic. It's adaptive behaviour, and it depends upon how we treat them and how they are housed. Not necessarily genetic, although I do feel that it is more than likely that there are some genetic implications for temperament as well. Alright guys, you made it through to the end. You must have if you're watching this, so well done. And Mr. DNA did want to talk about a lot of stuff that he's been following on social media and put his own slant on it. So once he gets going, you know what he's like, he goes on for an hour. So there you have it. I did actually leave a comment for Dr. Ben Morrell over at RGI and asked them about their sex testing from sheds. The answer that I got from him, and this is word for word, is that he has seen no convincing evidence that any species of snake is TSD, temperature sex determination, and ball pythons are certainly XY animals and genotypic sex determination. So that's Dr. Ben Morrell's answer to my question, which backs up my take on the TSD portion of this video. I do promise that the next videos will be a little bit more eye candy and a little bit easier to follow, probably a bit shorter as well. So thank you for wading through all that with me. Thanks for watching. Don't forget to share, like and subscribe, and we'll see you for the next one.